Hello. Hi, I'm Jayaram Kancherla. I work with the Center for Bioinformatics and Computational Biology at the University of Maryland, College Park. Something I want to talk about today is the file server. It's a system to query and transform directly on files. Just like a motivation or introduction to the tool itself. Data repositories like the roadmap epigenomics or the ENCODE project host a lot of large amounts of data as files. Some of the data comes from experiments such as bisulfite sequencing, so methylation data, gene expression, and so on. So given a repository and a bunch of files, let's say you want to calculate, like for example, in roadmap, you want to, in roadmap you want to calculate the difference in a differential binding in a particular histone marker for a particular set of tissues. Or if you're looking at the TCGA data for a particular cancer type, you want to calculate the average gene expression across normal and tumor samples. And so the current workflow to do this would be, so you would first download the files, use a computational environment like Bioconnector or some other thing, and then you would run your analysis, save the file, and then use a genome browser to visualize those results. And so our goal was to simplify that process. So what we do is, so we build this Python library called the Epiviz file server. If you have a limited number of files, you can manually import files, but given repositories have a large number of files, you can write a configuration file. I'm going to show a Jupyter notebook after this. So that's a list of files that are publicly available, and then you can define transformations of those files. And then you can, this library itself wraps everything into a REST API so they can build interactive tools on top of this. And you can use our Epiviz, which is a visualization tool from our group to visualize the data sets. We currently support bigwigs, bigbits, or any other files that can be indexed with tabix. I'm going to switch over to, sorry.
You found it?
Yeah, it's not, how do I make it full screen? So the configuration file looks like this. It has a location to where the file is located, what type of file it is, and then annotations that can be provided. So the way you can define a transformation is, so the first thing we do is you load all the configuration file as a set of measurements into the system. And then I'm filtering for a set of measurements that I want, and then you can define whatever computation that you want to do over those files. In this case, I'm picking all the H3K27M3 markers that have signal from the roadmap dataset, and then I'm trying to find the difference across the differential binding across those two files. And then you can use Epiviz to visualize those datasets. Just a quick tutorial. This is adding the raw signal in the beginning, and then the second track that you see is the differential binding that is calculated dynamically from the files. Something I forgot to mention was, so when you define a transformation, it is not pre-calculated. Once you're in a given range, it goes and gets the data from those files and then calculates the binding. So you can also embed these visualizations within Jupyter Notebook, and then that's just a quick demo of showing you could do that. How do I escape from that? I'm not used to.
Oh, where's it gone? Sorry. Go away, Chrome.
So the links to the Jupyter Notebook is available online. We also have another notebook that describes the library in much detail. If you're also loading files from Bioconnector, it has a resource called Annotation Hub. We have an easy-to-use function to directly import files from Annotation Hub. Just a quick introduction to Epiviz. So Epiviz is something that our group built out. It's an interactive visualization tool for functional genomic data. In addition to like track-based visualizations you would normally see, we support plot-based visualization. For example, if you're looking at gene expression data set across different markers or tissues, it's much more intuitive to look them as heat maps or scatter plots. We also implement brushing that links all visualizations by genomic region. The workspaces are shareable. And we also support, our visualizations are built on top of Web Components, which is a Web W3C specification. So you can embed them into any framework that supports HTML, so they're reusable and extensible. And all the links are available online. That's a GitHub repository. The documentation is available there. I want to acknowledge my advisor, which is Hector Corrado Bravo, and Yifan was an undergraduate researcher in the lab that helped us with some of the implementation. I'm also doing a poster, so P31. If you're working with a lot of genomic data files or trying to build tools on top of visualizations, stop by. Thank you.
I'm impressed he kept to time with a live demo and technical glitches. One more lightning talk.